Good day. This is Dr. Cywes. Today, we are going to discuss non-alcoholic fatty liver disease. We'll review the pathophysiology, the clinical manifestations, and then treatment from a ketogenic perspective. This talk is part of the launch of the Ketogenic Science of Therapeutic Carbohydrate Restriction textbook. Let's get to it. It is important to define the concepts of what fatty liver disease represents. Obviously, the commonest cause historically of fatty liver disease, no longer true today, is alcohol, and that's alcoholic fatty liver disease. In the modern era, we call it non-alcoholic fatty liver disease, and the first part is where fat accumulates in droplets, in little collections within and inside of the hepatocytes, or the cells that make up the liver. And that's fat accumulation. Then there are certain triggers of inflammation, both within those cells and in the blood vessels of the liver. And we now transition from what we call non-alcoholic fatty liver disease, which is just the fat accumulating within the liver, to now something called steatohepatitis, or inflammation of those liver cells. And that transition to inflammation is where scarring begins to happen. The early stages is reversible, but fat accumulation plus inflammation leads to necrosis or death of those liver cells, and progressively toward fibrosis of the liver and eventually cirrhosis, where scar tissue replaces liver cells. So we'll take you down that pathway and explain exactly how and why that occurs in the modern era and what the most common cause is. So as part of this, the liver is so central in human biology that I'm going to begin by explaining normal hepatic function, normal liver function in a demand system. Then we'll look at adaptations of those liver cells as you supply products to them. And then we'll also look at what happens to the blood vessels within the liver that have this very synergistic relationship with the liver cells themselves, and we'll look to see what happens to that blood vessel under supply conditions. Once we've identified and understood the causes and how the pathophysiology occurs, we can then help you as clinicians to treat non-alcoholic fatty liver disease and steatohepatitis, hopefully to complete remission. We'll talk about what the proper human diet is to achieve that, any medications and supplements, and also how to monitor your patients in identifying the disease and the disease transformation. So the human body is an incredibly, incredibly complex system, and nothing works in isolation. So it is very important that we have a systems approach to biology. And my entire way of thinking, and I would urge you to do this, is to stop primarily focusing your understanding and the work that you do on human outcomes data, but start looking at biology, at physiology and pathophysiology to understand and predict how your patients are going to behave. As I said, the liver is incredibly complex in terms of what it does. And if you think about the liver, it is the central functioning part of human metabolism. Everything traffics through the liver, and the liver and its soldiers, the hormones and other products that it puts out and takes in, are really the governing factors of your entire metabolic pathway. And I'm not even going to go through this complex picture, but as you can see, the liver touches every aspect of human metabolism. And in order to understand fatty liver disease, we have to be able to look at some of these processes, but also what's important to understand that once the liver becomes inflamed and cirrhotic, and even just inflamed, clearly these functions aren't performed very well. So it's kind of this self-fulfilling prophecy. Okay, folks. So here's a critically important aspect to understand about how the human body works. In terms of energy, the human body functions best and is designed to function as a demand-based system, okay? And so if you look at all the cells in the human body, the cells create or should create that demand. Now we're talking about normal. So under normal conditions, the cells create the energy demand, and it is the liver's job to supply that demand and keep pace with that demand using its hormone soldiers, and also by supplying the two sources of energy into the bloodstream. So what are those two sources of energy? Well, the sources of energy that most cells are able to use are sugar, glucose, and ketones, fat breakdown products. Some cells can only use glucose, some cells use both, some cells prefer ketones, and some cells, variably depending on their function, can either use ketones augmented by glucose. So when we're looking at what the liver supplies, and this is in the fasted state, the blood glucose and ketone concentration in the blood is one aspect. What is that number? And that number is very tightly controlled. And in the fasted state, it is controlled by the hormone glucagon. Now, the other part that most people don't pay attention to, because we always measure blood sugar and ketone levels, and we stick our fingers and we check what those... The other part that's critically important to understand is the flux of glucose and ketones through that system. In other words, how much, what's the grams per hour that enter the system and then get used up? And that's how we measure demand. And in a fasted state, that blood sugar and that ketone flux is supplied by the liver under the influence of glucagon when the human body is metabolism centered, breaking down other products to provide glucose and ketones to the cells under cellular demand. So the demand goes from the cells back to the bloodstream and therefore back to the liver. And if you look at the receptors for sugar in these cells, let's look at just glucose. All cells of the human body have GLUT1 and GLUT3 insulin independent glucose transporters. In other words, the uptake of sugar by most cells of the body is independent of insulin. Most people don't understand that, okay? So there is a constant demand and a constant uptake from blood. Think about this: when your heart stops beating, yes, it reduces oxygen, but when that flow stops, it's actually not just oxygen, but it's actually glucose flow to those cells that stops. And within a minute or two, the system can no longer function because of a breakdown in that constant uptake pathway. So all cells of the body take sugar up independent of insulin. Now, the two groups of tissues that have some of the highest demands for sugar are your muscles and your brain. And they have a slightly different glucose transporter in their cell membranes called the GLUT4 receptor, and that is insulin sensitive. And insulin can drive sugar up a gradient using that GLUT4 receptor. GLUT1 and GLUT3 typically are downstream, down a gradient. GLUT4 can be up or down gradient, and it is sensitive to insulin. In other words, insulin unlocks that gate, particularly in the muscles and the brain. And that's important to remember. So in the fasted state, all cells also, all nucleated cells have the capacity to store sugar as glycogen. And as cells in the body are working in the fasted state, they're creating this huge demand from the bloodstream to take up sugar, depending on if they primarily use sugar or ketones, and we'll discuss that in a second. But they also have the capacity to tap into their glycogen stores, although that is not desired. That's kind of their backup energy resource. So they use those glycogen stores. But when the cells demand energy, blood glucose to most cells is supplied through glucagon dependency, and then muscles and brains through insulin dependency. Now, the other fuel source that the body uses, and most cells can variably use preferably glucose or preferably ketones, the human body is primarily designed to preferentially use ketones as a fuel source. So by far, the majority of energy that a healthy human body uses is in the form of ketones, not sugar. And that's important to understand as well. And the supply of ketones to the bloodstream is glucagon dependent and insulin independent, okay? So when there's an energy demand by these cells in a fasted state, The liver is the primary metabolic resource that provides that energy. And at first, the liver releases its stored glycogen. The liver has a large capacity, about 400 to 600 grams of sugar can be stored in the liver as glycogen, and that's rapidly released into the bloodstream. But then the liver also can produce sugar at a fairly rapid rate, and that is called gluconeogenesis when it is produced from other sources. And the other sources are either fat or protein. And the glucose comes from a part of the fatty acid molecule or the triglyceride molecule called glycerol. And the fatty acids, a triglyceride is one molecule of glycerol and 3 molecules of fatty acids. The glycerol goes down the gluconeogenic pathway towards sugar, and the fatty acids typically get transformed into ketones by the liver. Now, the next critical point to understand is that in a healthy human body, protein is not a primary fuel source. Protein is a fuel source that is only really needed under starvation conditions. Protein is primarily a structural molecule that gets used for muscles, for tissue repair, tissue building, as a hormone. Protein is not a primary fuel source, although it can be used in the gluconeogenic and the ketogenic pathway as a backup, but not as a primary molecule in a healthy system. And that's a very important concept to understand. Now, in the fasted state, where do we really store massive amounts of energy? As I've said, we can store some as glycogen in the liver, but by far the majority of energy is stored in our fat cells or our adipocytes. And under energy demand conditions, a molecule which is low insulin levels, when the insulin level is low, all of this is happening. And under low insulin conditions, a protein called lipoprotein lipase, an enzyme in the fat cells, releases triglycerides, both free into the bloodstream as well as packaged in LDL, which are just transport molecules, depending on the length of the fatty acids in the molecule. Shorter medium chain triglycerides can be transported free in the blood. The longer, healthier fatty acids have to be transported, packaged in a transport molecule or vehicle, and we call that vehicle IDL and LDL. And the adipocytes release those triglycerides and LDL to the liver, where the liver transforms them into ketones for energy. Okay, so this is what this picture looks like. In a glucagon-mediated gluconeogenic liver, one that is fasted for cellular demand, the liver will use odd-chain fatty acids. It's interesting, it can't use even-chain fatty acids, even carbon fatty acids. Odd-chain fatty acids can become sugar. It recycles lactate, which is an energy byproduct from all cells. It really doesn't want to use, amino acids, but they can go in to the gluconeogenic system. But mostly it likes to use glycerol from the breakdown of fat. And obviously at the top here, you see a glycogen-rich liver. This is after a fed state. You can see that pink tissue is all staining, PAS staining for glycogen in the liver cells. And then it can release that into the bloodstream. And here you see a liver depleted of glycogen. We're going to come down to amino acids in a little bit in another slide, but it is important to understand that amino acids or protein is a backup, not a primary source. Now, the other critical thing is that through gluconeogenesis and through breakdown of glycogen, the liver produces a molecule called glucose 6-phosphate. And glucose 6-phosphate, that phosphorylated molecule attached to the glucose, traps it inside the cell. It cannot be transported out. And it's only under conditions of low insulin that phosphorylated molecule is cleaved, and then the pure sugar can rapidly rush out of the liver using one of the GLUT receptors. So it's the GLUT5 receptor. So in that way, the liver creates sugar from stored glycogen, from glycerol, a little bit from amino acids, a little bit from lactate, and some from odd carbon fatty acids. And that's how it provides at a very low level, okay? At a low concentration, provides sugar to the bloodstream. And that's important to understand, is that when sugar is created by gluconeogenesis, it stays at a low level. Okay, now, in that fasted state, in a fat-adapted body, which is the healthiest body out there. Remember, the human body, the cells of the human body preferentially use ketone bodies, not glucose, as their primary fuel source. So under these conditions, the liver can use fatty acids and, as a backup, certain ketogenic amino acids or protein through a process called beta oxidation, where if you go through all the biochemistry over here, really what the liver is doing is converting acetyl-CoA to two things. First of all, in large amounts in the fasted state, down here, under the influence of glucagon, HMG-CoA lyase produces ketone bodies, beta-hydroxybutyrate, and as a byproduct of that, acetone, and that's that beta-hydroxybutyrate that gets into the bloodstream that we measure as ketone bodies, that is biologically the primary fuel that almost all cells prefer to use in a fat-adapted, healthy human state. Now, the other cool thing to look at how beautiful the human body is, folks. The same process of beta oxidation under the influence of insulin, quarterback of the hormonal metabolic system, under the influence of insulin, HMG-CoA gets reduced by HMG-CoA reductase under the influence of insulin to become cholesterol. And that is how about 2/3 of the cholesterol of the human body is created or generated in all the cells, but particularly in the liver. The liver is a massive factory for cholesterol, which is a vital molecule that governs every aspect of every cell's life. The cell membrane of every cell has to have cholesterol in it. And cholesterol is the precursor for all of the steroid hormones. And look here, folks. Not only is the creation of cholesterol under the influence of insulin, but also the first step of the pathway, the enzyme of cholesterol downstream to the steroid hormones that govern metabolism, is also controlled by insulin. And it's controlled by insulin at a genetic level, where high levels of insulin reduce those enzymes. And again, as you can see here, glucagon triggers ketone bodies and insulin blocks that to a certain extent. So this is healthy ketone and cholesterol synthesis in a healthy fasted state. So we've got glucose generation, gluconeogenesis, we've got ketone generation. And here's the part that we really need to understand properly. Because as we try to convert people to eating more animal products, we can screw that up pretty badly with bad advice. One of the critical things to understand is that glucose in the human body is primarily used as fuel, with just a few parts of glucose being used as a structural molecule. Ketones are both fuel and structural. Large amounts of fat in the human body in all cell membranes in a variety of different places. So ketones, cholesterol are used structurally, but ketones are also the primary fuel source for the human body with glucose as the backup. Protein, folks, is primarily structural. So glucose primarily energy, protein primarily structural. Glucose has a few structural functions. Amino acids, protein, has very, very little in a healthy human body energy supply. Now, of the 21 or 20 amino acids that we have, some are glucogenic. When they get processed in the gluconeogenic pathway, they turn into sugar. Others become ketones, the ketogenic ones, as you can see in this graphic. And some amino acids have both properties. But here's the problem, folks. When you eat a lot of lean protein and the human body is somewhat starved for sugar, especially if it's not fat adapted, and fat adaptation means that the human body preferentially uses ketones as opposed to sugar. If you still glucose adapted, glycolytic, if you still depend on sugar as your primary fuel source, and you're not consuming enough sugar or in that fasted state, now your body uses protein that you ate and also your muscles and break down protein products from the human body. Remember, the human body has no capacity to store protein. We'll get back to that in 1 second. So when the human body, in a pathological way, breaks down a lot of amino acids to provide glucose or ketones, the byproduct of that excessive protein breakdown, the protein byproducts, excrete uric acid and oxalic acid. And those are the two dominant sources of crystals that go to the kidneys and crystals that go to your joints. And we call those diseases calcium oxalate crystals, calcium oxalate kidney stones that can destroy your kidneys. And also we call uric acid in the joints gout. Gout and kidney stones, oxalic acid kidney stones, are primarily the consequence of a dysfunctional, imbalanced diet where we are using protein in a place it's not supposed to be used as a fuel source rather than as a structural protein. And as you become fat adapted, as you use fat as your primary resource, gout and kidney stones go away. You may have a flare while you're fat adapting, but to put gout and kidney stones into remission in your patients, it's very simple. Protect the protein with adequate fat. So a high-fat, ultra-low carbohydrate, moderate protein diet is the way to prevent those two diseases. And that occurs in the liver as a source of energy production. In fact, it's interesting if you think about it, the body has massive capacity for fat storage. That's what obesity is. The body has moderate capacity for glucose storage in the liver and in almost every cell. The body has zero capacity for protein storage. All protein in the human body is actually being used. Obviously, our muscles being the greatest depository of that, but your muscles have other functions. Think, people, biology. Okay, so we understand what a healthy, normal, functioning liver looks like in a fasted state, and that is a demand-based system, and those are systems where insulin is at a very low level, insulin and glucagon working very closely together, but glucagon is the primary driver of that energy supply system. Now, obviously, we're not a self-contained unit, so from time to time, we have to supply energy to the human body. So let's look at how the liver functions under conditions of healthy energy supply. On a low carbohydrate, moderate protein, high fat diet, let's see what happens. Under these conditions, as you eat, even if you don't eat a lot of sugar, your blood sugar levels do rise slightly, imperceptibly slightly from that fasted baseline. And now we switch over to insulin control. So what happens is as you eat, hormones in the gut, for example, GLP-1 in the upper intestine gets released that triggers the pancreas to say, hey dudes, there's an incoming load of calories coming in, and if there's any sugar or protein in that, we've got to trigger insulin. So the pancreas preemptively secretes insulin. Remember, the glucose and protein gets taken up through the blood vessels in the intestine and goes directly to the liver. So there's a very high, what we call first pass effect in the liver. The liver has this filtration system called a portal venous system, where almost all of the blood from the intestine first flows through the liver. And obviously the liver has major effect on impacting what comes in, processing that, and changes what goes out of the liver to the rest of the body. So the liver controls almost all of that income. In fact, the only thing the liver really doesn't control is long-chain fatty acids and fat-soluble vitamins. Be that as it may, even that is controlled by the liver through bile. We'll talk about that in a second. Be that as it may, when you feed, now you switch from glycogen-dominant release from the liver to insulin control of what you're supplying to your bloodstream. And technically, under a healthy insulin-sensitive state, your blood sugar goes up imperceptibly slightly, but it shouldn't go up very much because insulin should be very on top of that. And insulin gets primarily the liver to suck up a huge amount of sugar. Insulin also encourages those fasted state cells, particularly the muscles in the brain, to suck that up and also drive some sugar into the cells, although it may be gradient dependent, where all cells that have nucleuses store some of that sugar as glycogen. And also what insulin does, obviously, it switches off glucagon and switches off the release of ketones and sugar into the bloodstream. It also switches off the fat cells releasing fat. Now, under those conditions, our blood glucose ketone flux changes. So our blood sugar goes up, our ketones go way, way down, not to zero, but way, way down. And the amount to which the sugar rises is still controlled by cellular demand, but now the cellular demand by the liver cells is much greater. But cellular demand still controls blood sugar and the flux of sugar. So very rapidly, the body distributes what you've just eaten into all of your cells, some directly and some through process by the liver when that energy is supplied by the mouth. And this is what we call storage-centered as opposed to metabolism-centered process. And there should be an ebb and flow. Now, let's look at what this looks like. So, as we said, in the fed state, the liver changes from being the net provider of energy, and the energy is now provided, energy supply comes from your mouth, a little bit of glucose, mostly fat, and a moderate amount of protein. That is the healthiest diet you can eat. And the liver processes that. What the liver is doing is, number one, storing some of that sugar as glycogen and taking some of the protein and converting it to glycogen and storing it, but at a very low rate. The liver is also, and this is critically important, under the influence of insulin. Under the influence of insulin, the liver is not only turning excess protein to sugar, but the liver is creating body structural proteins. So those amino acids, those building blocks come in, and insulin has an incredibly powerful role on protein synthesis that happens during and after a meal. So insulin governs every aspect of incoming metabolism. So your glycogen stores get filled up, your protein production, structural protein, happens in the liver, and any excess short-chain fat and sugar is converted by the liver once the glycogen stores are full to fat, to triglycerides, and some of the shorter triglycerides that come from glucose, the palmitic acids, the oleic acids, the 16 and 18 carbon structures of saturated fat, are small enough that they're soluble in water. So those triglycerides, as we can see down here, are released into the bloodstream, just attached to globulin or albumin, and they're transported free in the bloodstream, and we can measure that when we do blood levels as blood triglyceride levels. On the other hand, if there's any longer chain fatty acids or complex fatty acids, polyunsaturated fatty acids, long chain monounsaturated and saturated fatty acids that are longer than about 20 to 22 carbon length chains, those get packaged by the liver into this large cholesterol-containing molecule called VLDL. And VLDL, early VLDL or juvenile VLDL, gets transported into the bloodstream, combines with HDL, and becomes the transport molecule that transports long-chain fatty acids, cholesterol, and some proteins from the liver to the fat cells. Follow me along? So it transports that to the fat cells, and during that time, and as part of that degradation in the bloodstream, VLDL doesn't live for very long. In the bloodstream, that gets transformed into a molecule we all know so well called LDL, which is the stable transport molecule of fat from the liver to the fat cells. So during this metabolism of food, the liver is storing stuff as glycogen, turning food into fat, transporting that out, and releasing sugar into the bloodstream, so that for a while, that sugar is, the human body is sugar dominant and the ketone levels are relatively low, although not zero. Now, at the fat level, at the adipocyte level, the fat cells are storing the fat as triglycerides, but there's also what we call de novo lipogenesis. The fat cells also have a very low capacity to turn sugar into fat, and that occurs in the fat cells. So just a wonderful, sensitive system, complex system that interplays really nicely when you supply the liver and the human body with real food. A little bit of glycogen, a little bit of glucose, mostly fat, and some protein. So here's what happens. The larger fat that we eat, the big chunks of fat, those long triglyceride molecules, are not able to be transported in water, in the blood, because they're too big. So the cool thing is here from the gallbladder, the liver, and actually in the liver produces bile, which gets squeezed into the intestine, and that bile forms little soap bubbles that suck up all of the big, long fatty acids that we eat, and the triglycerides from our diet, as well as the fat-soluble vitamins. That's why it's important to consume your fat-soluble vitamins with a fatty meal. Taking them without that, you can't get absorbed. And the molecule that gets produced is called a chylomicron, and the chylomicrons do not get transported in the bloodstream. In the portal venous blood, they get transported in the lymphatic system, and they dump into the systemic vascular system, not through the liver, but directly to the cells. They get transported to the subclavian vein on the left side, through the thoracic duct, which is a lymphatic channel, and they then enter the bloodstream, and they go to the fat cells. Now, in the fat cells, as your insulin level goes up, it blocks this protein called lipoprotein lipase from breaking down fat and releasing it into the bloodstream. And in fact, lipoprotein lipase then becomes a net absorber of that fat, and the fatty acids are then removed from the chylomicrons and stored as triglycerides and maybe phospholipids in the fat cells. In addition, some of the chylomicrons obviously circle through to the liver. And those chylomicrons and the breakdown products, the remnants, the empty chylomicrons, under the influence of insulin, enter the liver, where cholesterol is made, where protein is made, and under the influence of insulin, the sugar is taken up and turned into triglycerides. Now, the sugar becomes free triglycerides that also flow to the fat cells, free in the blood, and we measure that as triglycerides, where they get added to the fat cells. In addition, the liver takes longer fatty acids, packages them in VLDL, and discharges them into the bloodstream as VLDL plus HDL, which becomes your LDL-A, or your big fluffy LDL, packaged with a lot of triglycerides, some cholesterol, some protein, as a delivery system, as a vehicle that goes from the liver to the fat cells. And in fact, and here's an important step to remember for the next section, the rate-limiting step in the liver, the rate-limiting step of de novo lipogenesis, of turning sugar into fat, is not the production of the fat, but it is the packaging and the transport of that fat in the liver cell to the bloodstream. That's where the backup is. The backup is in the packaging and the creation of LDL and VLDL. So if that's the backup, under conditions of high sugar uptake, more and more of that sugar just gets converted to palmitic and oleic acid, which are water soluble, and get transported as triglycerides. So under those backed up conditions, the way the liver deals with that is to put more and more of that fat that it's created as triglycerides into the bloodstream, and also your LDL count goes up. And the variability of that is determined as the rate limiting step of LDL production, based upon the rate at which the liver produces fatty acids. And by the way, folks, that step is insulin sensitive. Insulin at a genetic level controls the SREBP, the enzymes that are responsible for that pathway at a translational level. So insulin has an influence on the membrane uptake of sugar, but also has a very, very powerful influence on the genetic production of the enzymes that produce cholesterol and that produce LDL. So insulin is a quarterback or a central figure in this entire step. But if you're eating a low carbohydrate, high fat meal, this system works extremely well to replenish stores. Now, that's the way it used to be, folks. That's the way it used to be when, as a species, we human beings had a scarcity of food. In the modern era, over the last 100 years or so, we've transitioned from a race of scarcity or a species of scarcity to a species for the first time is encountering abundance. And because we are heavily driven to consume huge amounts of food, now we have entered the realm of oversupply. Now, I'm just going to return back to the slide for 1 second. If we eat a lot more than we need, because this is the rate limiting step, some of that fat remains behind in the liver cells. And in the fed state, especially the slightly excessively fed state, on a low carbohydrate, high fat diet, there is a little bit of fat accumulation in the liver that then becomes used for ketone production and slowly transported out of the blood system. That's why our LDL-A particles go up and our triglycerides may or may not go up, depending on how much we can get into LDL versus triglycerides. Okay. Now, in the modern era of energy oversupply, now we're dealing with this massive constant consumption of a high carbohydrate, low fat diet. And your blood sugar levels rise very, very rapidly, especially when we're eating simple sugars. You drink a Coke, you eat some ice cream, simple sugars that have a very high glycemic index, not the glycemic load, not the amount, which also counts, but very rapid rise in portal venous blood system, in the portal venous blood system, and the body cannot produce insulin fast enough. And this is called insulin resistance type 1. The body is still producing insulin, but it's not producing it fast enough to cope with the amount of sugar that you've produced. And this is insulin resistance type 1. So your blood sugar actually rises up and to the upper limit of normal or maybe a little bit higher. So that is measured on a finger stick. But also, there is a massive amount of glucose flux. The number of grams of sugar, the glycemic load, is enormous in the blood system. And the glucose flux, the amount of sugar in the bloodstream, that glycemic load, is controlled by your brain and your hands stuffing your face. So it's supplied by the mouth in the form of meals, high carbohydrate meals, snacks, sugary drinks, continuous consumption. And here's the problem, that the human body over time now goes into defense mode. Because one of the things to understand is that chronically elevated level of sugar is highly, highly toxic to the human body. And it's toxic primarily in two settings. The first part is it is toxic in the bloodstream. So when your blood sugar goes up, that is toxic to your red blood cells. And how do we measure that toxicity? Through a blood test called hemoglobin A1C. Hemoglobin A1C is just the first measure of sugar toxicity to the cells, the red blood cells and the hemoglobin in the blood vascular space. But it also, as we'll see in a little bit, affects the endothelial cells that line the blood vessels. I'm going to come to that in a second. So sugar is very toxic in the bloodstream, and the human body is very, very heavily geared and has a number of backup systems to rapidly remove sugar from the bloodstream. It gets taken up by all cells where it gets converted by glycogen, but now we've maxed out that glycogen. And it gets taken up by the liver where it's stored as glycogen and it gets turned to fat. But in the blood, if you're not able to remove the sugar load quickly enough, either because of profound insulin resistance, where cells are just not taking it up, or because the supply is continuous and excessive, that causes vascular injury and injury to the elements in the bloodstream, which are your white blood cells, your immune cells, your hemoglobin, your oxygen carrying capacity, and your hemoglobin, and the cells that line the blood vessels. All of those cells get injured, and we'll come back to that in a little bit. That is called diabesogenic disease. However, the other part is if that sugar rapidly gets distributed into the cells, those sugars get trapped in the cells by phosphorylation. The first thing that happens is glucose enters those cells, and under the influence of insulin, you get a phosphate molecule being put onto that glucose, and now it can't get out. So it's trapped inside of the cell. And that sugar is still very hydrophilic. It takes with it a molecule of water. So the cells swell up, and they don't function that well. So intracellular hyperglycemia, which is almost impossible to measure, we can see it in the lab, but very difficult to measure clinically. Intracellular hyperglycemia is extremely damaging to the cell. And we're going to look at the liver cells to see how that happens. And whether you clear sugar rapidly from the bloodstream and get intracellular hyperglycemia, or whether you develop vascular hyperglycemia that can be measured as a blood glucose concentration, is determined by insulin production capacity, and that's genetic. But it's also determined by the amount of simple sugar and sugar that you eat. And under these conditions, the vascular and cellular system is now in defense mode. It is defending itself from injury, but bad things happen. So let's have a look at this. First of all, the body, when the sugar rises in the bloodstream, this energy oversupply of glucose. And remember, the other thing that's critically important, that almost always, along with massive loads of glucose, in the modern era where saturated fat is bad for us, comes a whole load of polyunsaturated, hydrogenated, or oxidized polyunsaturated fatty acids. And those are the fatty acids that come from the seed oils that are heated out of our products. And very often, high sugar, especially the simple sugars, are packaged, not always, but packaged with polyunsaturated fatty acids. Think donuts, okay? So this massive energy supply of not only sugar, but polyunsaturated fatty acids has to be dealt with. It rapidly enters the bloodstream, and it gets distributed to all the cells, but very quickly, the cells say, dude, you're hurting me here. My intracellular hyperglycemia is killing me. And the cells do their best to block the GLUT1, GLUT3, and GLUT4 receptors. They phosphorylate those receptors. They shut them down to close the front door so that glucose can't get in, which then causes a buildup of sugar in the bloodstream. Now, if you're a high insulin producer, you can produce massive amounts of insulin that still force that sugar into the cells, and your blood sugar stays at the upper limit of normal. It doesn't mean that there's not a problem, but we call that insulin resistance. When the sugar builds up in the bloodstream and you can't get it into the cells because you can't produce enough insulin, we call that prediabetes. It's measured similarly, but there's a different effect. Insulin resistance occurs with a mild to moderately elevated vascular hyperglycemia, but a massive intracellular hyperglycemia. That's insulin resistance, and typically those people typically do not develop diabetes until it's very, very late. On the other hand, prediabetes is the same issue, but here your blood sugars are much higher, and you can't get the sugar into the cells. And it's an important concept to understand that normal to slightly elevated blood sugar with high insulin level is insulin resistance, not diabetes, intracellular hyperglycemia, whereas high blood sugar with moderate insulin levels is prediabetes, or diabetes, frankly. But we'll talk about that again in a second. So now you've got this massive continuous uptake of sugar by the liver cells. And what's interesting, folks, is this. The hepatocytes, the liver cells, take the most of their sugar up directly from the bloodstream. It's the only cells in the body that have direct contact with the bloodstream, and they suck up, they're like sponges for the sugar. So they rapidly increase their glycogen supply. And I call glucose and polyunsaturated fatty acids not food, I call them drugs because they are toxic to the human body. So the liver maxes out its glycogen supply. It traps the sugar in the liver cells as glycogen 6-phosphate and fructose 6-phosphate. Fructose and glucose have the same phosphorylation process, and it rapidly turns that sugar into fat, into fatty acids. It adds glycerol, and it produces a ton of triglycerides. And as it produces those triglycerides, obviously, they accumulate in the liver cells. Folks, that's called fatty liver disease. Non-alcoholic fatty liver disease is related to a glucose energy oversupply where the liver is trying to cope by turning that sugar, the toxic hygroscopic sugar, into non-harmful fatty acids. The fatty acids are neutral from a toxicity perspective. And the way that the liver cells protect themselves and the body from damage is by converting toxic sugar to fatty acids. The fatty acids are not by themselves harmful yet. So fatty liver disease, non-alcoholic fatty liver disease, is a protective mechanism protecting against what your face has done by stuffing sugar and starch into it to your body. So fatty acids accumulate in the liver cells as triglycerides and being packaged into VLDL and reprocessed LDL and possibly chylomicrons. But the problem is, remember from the previous slide, the rate limiting step is the movement of that fat from the liver cells into the bloodstream. So it's accumulating in these vacuoles, in these collections, and that's visible at biopsy in the liver. And it's also visible on ultrasound to see this big fatty liver, because there's a certain look to that fatty liver. Now, as that liver becomes resistant to insulin, cholesterol production, protein production, both of those go away. So now your protein is actually entering the gluconeogenic pathway, and those byproducts of oxalic acid and uric acid are causing gout and kidney stones, because insulin is no longer able to drive the transcription of protein synthesis, nor cholesterol. So now you're getting these packages of VLDL and LDL that are deficient in adequate cholesterol. See how insulin is so central to this? But also, insulin no longer is able to block glucagon. So now you've got glucagon functioning independently of insulin to convert the protein to sugar and to release this excess sugar into the bloodstream to dephosphorylate that G6P into glucose and shove it back into the bloodstream. And the bloodstream's already over capacity with sugar. And that's called paradoxical gluconeogenesis. The liver is desperately trying to cope and stay alive. And under those conditions, that's where your blood sugar goes up. And if you're a low insulin producer, we call that disease over time diabetes. Now, on the other side of that, the polyunsaturated fatty acids and some of the fatty acids that get produced in the liver have a lot of double bonds. And those double carbon bonds are very unstable, particularly when they're amassed in the vacuoles of the liver. And they break, and when those double carbon bonds break, they release oxygen-free radicals, hyperoxide, peroxide. They release all of the free radicals. And that is what we call, folks, inflammation. And if you think of antioxidants, it's designed to try to control that reaction. So the polyunsaturated fatty acids, who by definition have a number of different double bonds that are collected in the liver, break apart. And that is where you now transfer from this protective creation of triglycerides and fatty acids to an inflammatory model. And now that fat is no longer neutral. It is actually the source of inflammation in those cells. And we call that NASH, or non-alcoholic steatohepatitis. And we see those as a foaminess. Instead of these big vacuoles of fat, it now looks like foam. And that is a highly inflammatory condition in the cell. And guess what, folks? Under the conditions of that inflammation, the liver cells can't work that well. And the situation now gets out of control, and it's this positive spin. And that is where those liver cells die. There's one other element that we're going to talk about in a second, but that is where those liver cells die or get damaged. They release AST and ALT into the bloodstream. So under conditions of steatohepatitis, if you're going to measure this with in the blood system, look at your alkaline phosphatase, look at your AST, and look at your ALT, and those numbers rise up. When they get into the 20s or higher, that's telling you that there is intrinsic hepatocellular damage from inflammation. So you've got fatty liver now transitioning to steatohepatitis, and it is measurable in the bloodstream because those damaged products, those enzymes from the liver being released from damaged liver cells into the bloodstream are measurable. So now that inflammation of both the sugar as well as the polyunsaturated fatty acids causes steatohepatitis inside of the liver cells. Now, the adipocytes are still trying to store all this fat and trying to create some of the fat from the sugar, de novo lipogenesis, but the transport to those fat cells is enormous and continuous. There's no ebb and flow. And folks, the other disease, the way we see that disease is as obesity. That's how obesity happens, with this massive, chronic, excessive consumption of carbohydrates, conversion to fat, transported as free triglycerides. So your triglyceride levels in the bloodstream goes up. Also, your LDL goes up if it's possible, and you get high responders and low responders based upon cholesterol production. So that explains the LDL part, and then that gets transported to the fat cells. So here we go. The mechanism of fatty liver disease and non-alcoholic steatohepatitis has nothing to do with the consumption of fat, except that maybe it's too little. What causes insulin resistance, what causes fatty liver, is the way the liver copes with chronic excessive carbohydrate consumption. And chronic excessive carbohydrate consumption, like alcohol, is a substance abuse problem. I am the Carb Addiction doc. I've got a whole series of lectures on why sugar consumption is not a food consumption problem. It is a substance abuse problem. And look at what happens to the brain under those conditions of substance abuse. It lights up as if you took a hit of heroin. So fatty liver disease is a disease of the human brain, addiction, and the human mouth, consumption of sugar. And it is a biologic response, it's a pathophysiologic response to defend against chronic excessive carbohydrate consumption. And when you add polyunsaturated fatty acids, an accumulation of that double bonded fat in the cells, that produces the inflammation that is now causing steatohepatitis. And in particular, it is fructose, glucose, and lactate that form these triglycerides, and when the glycogen stores are full, form this fat with a rate-limiting step of eviction from the liver to the fat cells. As long as you can get those fatty acids and triglycerides to the fat cells, you become heavy, and it dampens that steatohepatitis effect. But when that transport system is broken out of the liver, and again, it's insulin-mediated, that's where the inflammation occurs in the liver. So it's excessive consumption. Okay, and here we see that picture. In a normal liver, in the fasted state, you've got glycogen depletion. Very pretty, normal-looking liver. In the fed state, you've got huge accumulation of glycogen, which is this pink substance. It's a PAS stain. The other thing also, folks, to look at over here is that under healthy conditions, every hepatocyte is directly connected to the bloodstream. The blood is so important in that liver that every hepatocyte is bathed on multiple sides directly with blood. Come back to that in a second. Now, under pathologic conditions, you've got all these fatty vacuoles collecting. You've got the fat collecting. Okay? And if you notice, as those livers swell, guess what they do? They compress the sinusoids, and the sinusoids, as you'll see in a minute, also get blocked. Those are the sinusoids are the ones that bring blood to the cells themselves. They're like the little capillaries of the liver. So look how swollen this is becoming. And that's a fatty liver with swelling and edema. And then on top of that, you get hypoxia, lack of oxygen and lack of nutrients because the vascular supply is affected. But also, you get the release of those oxygen-free radicals. And now you see these blue cells over here, folks? Those are white blood cells. That's inflammation. That is steatohepatitis, or non-alcoholic steatohepatitis, inflammation of the liver, and that's what kills the liver. That kills these cells die, and they're replaced with fibrous tissue. That is cirrhosis. And that's how the liver goes from being partially damaged or reversibly damaged to being irreversibly damaged. So one other concept we have to understand, and this will make the entire biology of carbohydrate consumption understandable in the whole body, but it's particularly important in the liver as it pertains to steatohepatitis. Steatohepatitis doesn't just happen in the liver cells themselves, but under conditions of energy oversupply that we've just talked about. Here's what happens. Sugar is very hygroscopic. So when you add sugar to water, the sugar actually dissolves in the water. So when you look at this in a blood vessel, these are the cells that line the blood vessels, called endothelial cells. And when you add sugar, high concentrations of sugar, these endothelial cells take that sugar up in an insulin-independent way. And they swell up like toads because every molecule of sugar is attached to a molecule of water. So the sugar enters the cells and the cells swell up. So in fact, if you look at healthy endothelial cells, they should look like a fried egg, okay? However, when those fried eggs swell, they turn from being a fried egg to being more like a boiled egg over here. And you notice the gaps, you notice the spaces in between. And those gaps are now the surface of the liver, of these endothelial cells. This surface is anticoagulant. Things don't stick to it. But now when those cells ball up and they leave little gaps between them, that then has to be dealt with through activation of the clotting system, because now you are directly exposing cells of the body to the blood elements, and that may not be a good thing. So again, as we said, under normal conditions in the liver, you can see every liver cell is bathed in a sinusoid, lots of blood liver cells interaction. Okay, lots of, because that's what the liver cells do. But here, as they swell up, you can see the foaminess in these cells. That white stuff is all foam in these cells. That's fat, folks. That's inflamed fat. And guess what? Look how compressed those sinusoids are. No blood flow, clotting activation. And you know, when you don't get blood or oxygen downstream, things don't work so good. And that's what causes the inflammation that kills these cells. So let's look at this in a schematic. In a normal fasted state, the endothelial cells up here are nice and flat like fried eggs, and they're touching. And you've got a wide lumen, lots of flow coming into contact with the hepatocytes, just beautiful transition of metabolism, doing very well. Then after a high carbohydrate meal, glucose enters these cells and they round up. First thing they do is they compress the lumen. So now you've got back pressure to the radius to the fore. That's hypertension in the rest of the body, but it causes hepatic hypertension, portal venous hypertension. Portal venous hypertension, intrahepatic portal hypertension. For those of you who have studied the liver, you understand what that is. And that in large part is triggered by these endothelial cells swelling. The other thing they do is they disconnect and they expose directly much larger pieces of the liver. And this is called the space of Dis. The liver does not have a basement membrane. And now, this is my Ph.D., folks. I looked at this injury. I caused this injury. How did I cause it? By flooding the liver with sugar. No magic to that. Within 3 hours, I could create these little fibrin plugs. And my Ph.D. was looking at platelets that then get activated and enter that little fibrin plug. And then if that plug doesn't dissolve, which they often do in that space of Dis, now you're getting the infiltration of Kupffer cells, of macrophages, of white blood cells that are highly activated. That's the inflammatory system that's getting activated, not in the liver cells, but feeding the liver cells. And that vascular inflammation attracts more clot, not so much in the liver, but in other places, you're now getting this lipid cholesterol plaque laying down. That's what LDL and triglycerides do. And that's why they have an integral part in the late clotting system. And that's why they've been mistakenly maligned in the lipid heart hypothesis of cardiovascular disease. Be that as it may, in the liver, inflammatory platelets get activated. They clot and block the sinusoids, causing ischemia and hepatocellular death. And biologically, at least and pathologically, when you look under the microscope, we call those foam cells. And that is the combination of intracellular inflammation and vascular inflammation that leads to irreversible non-alcoholic steatohepatitis. Now, this is from my PhD. This is what a normal liver looks like, and it's beautiful. The liver obviously sees a lot of blood. This is the sinusoid. This over here, for example, is a nice fried egg endothelial cell, and you can see it's got little gaps to it. We call those fenestria windows, where there's direct exposure to the space of Dis, or this underlying little area between the liver cells underneath the vascular cells, so that the liver cells can directly come into contact to remove and replenish the blood supply as it performs metabolism. Here we see a blood vessel, there's an endothelial cell, and there we see the space of Dis. Very, very healthy. Now, folks, let's add sugar. This is from my PhD, electron micrographs. So we add sugar, and the endothelial cell balls up, becomes this boiled egg, and it exposes all of the microvilli over here. Well, dudes, I don't like to be exposed, so let's activate the clotting system. And now in this bottom picture over here, you can see some early platelets coming in. These are activated platelets over here, sticking to this blood vessel. There's your liver cell, sticking to these liver cells. And the activated platelets form clumps. The clumps begin to plug those capillaries. Here you can see a beautiful capillary, and here you've got red blood cells trapped, you've got platelets trapped, you've got fibrin, you've got white blood cells, Kupffer cells, just the snot that is lying in the blood vessels, completely blocking that blood vessel. And guess what? No energy, no oxygen. These cells here, that is a very sick, dying hepatocyte. That, folks, is the start of fatty liver cirrhosis. So, as we said before, the human body is just a beautiful thing. And in response to chronic excessive carbohydrate consumption, based upon your human genetics, the body responds in slightly different ways. It's got to defend itself under conditions of chronic excessive carbohydrate consumption. And in certain people who cannot produce a huge amount of insulin, people over here on this bell curve that cannot produce a lot of insulin on the left of the bell curve, those people have very early insulin resistance. And the insulin resistance by the cells of the body prevent those cells, protect the intracellular hyperglycemia from happening, protect the inside of the cells by blocking sugar from going from the bloodstream into the cells. Therefore, your blood sugar builds up. You see insulin resistance, mild elevation if you measure insulin. And I would urge any doctor working in this space, which most doctors never do, is to check C-peptide and insulin levels. And if it's moderately elevated, but you've got a very high blood sugar, you know that person is diabetogenic. And the diseases you can expect would be a rise in A1C that we call diabetes, type 2 diabetes. And you see moderate to high elevated triglycerides as well as LDL, because some of that sugar by the liver is still being turned into fat, but not a huge, awful lipid problem. But we see low HDL, high CRP, C-reactive protein, because that elevated sugar in the bloodstream is activating the clotting cascade and activating the inflammatory system. We see an elevation in white blood cell count. We see an elevation in ALK-phos, gamma-glutamyl transferase, and a moderately elevated AST and ALT. We see a degree of fatty liver, but not awful. But all of the damage is happening intravascularly, and that is where that clotting cascade that we just described is being activated throughout the body. And diabetogenic low insulin production is a vasculopathy. It is where the disease occurs mostly in the blood vessels, and it is an oxygen and food supply blockage. And the diseases we see, type 2 diabetes, mostly fatty liver disease without a lot of fibrosis, unless there's a lot of polyunsaturated fatty acids being consumed. We see atheromatous cardiovascular disease ramped way high. And here's the bizarre part, folks, is that often that atheromatous disease is not represented by massively high triglyceride or LDL counts. Yeah, they can be elevated, but they don't have to be to have this atheromatous disease. You see hyperglycemia, you see massive edema, interstitial space edema, you see hypertension, we see endothelial inflammatory cells, we see some overweight, but not massive obesity, and we see paradoxical gluconeogenesis. On the other side, we have people that can produce massive amounts of insulin. Again, by measuring a C-peptide and insulin, these people will have a 5-6-fold increase in their insulin C-peptide numbers. Not out of the normal range, because remember, the normal range is based upon the standard American diet criteria, but for someone in a ketogenic diet. And we'll discuss that in a second. These people are obesogenic. Their blood sugar may not be that high. Upper limit of normal, slightly above 100 in milligrams per deciliter. The A1C may be elevated, but not in the diabetic range. They have very high insulin C-peptide, usually very high triglycerides and LDL because they can produce a lot of fat, low HDL, relatively non-inflamed vasculature. White blood cell count not horribly raised, but mild to moderate elevation of ALK phos and elevated AST and ALT. This is where the intracellular steatohepatitis is increased. These people have obesity, they have steatohepatitis, massive obesity. They have steroid hormone diseases. Their risk of inflammation, their risk of cancer, their risk of Alzheimer's disease, their risk of autoimmune disease through the roof. They have hypercholesterolemia, and they have an intracellular inflammatory process. And they lean more toward the obesogenic side. Not entirely in the scope of this discussion, but important to understand as it pertains to the liver and fatty liver disease. So, folks, when it comes to changing our perspective, we've got to understand that the human system, this complex human system, is an energy demand system. And that energy demand is driven by the body cells. Human energy balance depends on the ebb and flow of energy to and from the liver based upon intermittent fasting and feeding, aerobic activity that drives physiologic hunger. And it's interesting that if humans are genetically designed to be in ketosis most of the time, which we are, healthiest place to be, then the assumption that insulin regulates glucose metabolism has to be false. Because that, under ketogenic environment, is the responsibility of glucagon. Insulin only responds to glucose supply, but in excess, that has toxic consequences, particularly under conditions of insulin resistance, and that's measurable. Hepatic glycogen is the first responder to energy demand, release of glycogen from the liver, but our fat cells are generally developed as energy reserves, this massive store of energy. And humans are designed to be in ketosis. Therefore, fat cells are not the recipients. Fat cells are the recipients, not the drivers of obesity. The liver is the driver of obesity by turning sugar into fat. It is also important as you counsel patients about what to eat, don't overeat protein by itself. Protein is a nutrient, not a primary fuel. And if you're eating enough fat and you're fat adapted, you don't need a large amount of protein, because fat is the fuel, not protein. Now, if insulin regulates fat and cholesterol metabolism and protein synthesis, then insulin regulates, as we said, all of the metabolism of the liver. Excess carbohydrates get converted to triglycerides, LDL and VLDL as transport molecules, and that's an insulin effect, trying to cope with chronic excessive carbohydrate consumption. So don't blame the lipids for disease that's caused by sugar. Lipids are innocent. Lipids are innocent in this process. Systemic harm, therefore, is not due to dyslipidemia. It is due to chronic excessive carbohydrate consumption, vascular injury, and intrinsic and clotting and intra and extracellular inflammation, and all organs are affected. So as we see in somebody who is a large carbohydrate consumer, whether it's apples or ice cream, doesn't matter how that sugar goes into the bloodstream, at first, what happens is after every meal, your blood sugar goes up, insulin gets released, blood sugar drops, and you get hungry. So now you're going from 1 or 2 meals a day to 6 to 8 meals a day. You're basically intermittent eating in a high carbohydrate diet. And to cope with that, your pancreas produces more and more insulin and C-peptide, which reaches the genetic maximum of insulin production. We call that insulin resistance stage 1. Blood sugar is upper limit of normal, and the body is transitioning all of that sugar into massive amounts of fat, but that elevated insulin blocks the conversion of fat release and blocks the production of ketones. So these people are in glycosis, and as long as that system is functioning okay, all of that sugar gets turned to fat, and those people are obesogenic, enormous, but spared the ravages of diabetes. And those folks get fatty liver disease. Now, in insulin resistance stage 2, this is where your glucose consumption is in excess of maximum insulin production. So now, instead of insulin controlling blood sugar to a certain extent by removal into the cells, particularly liver, and transition to fat, now your blood sugar begins to rise. Your insulin level is maxed, your blood sugar starts to rise, and glucose conversion to fat is blocked because it can't get into the liver cells adequately, and you have paradoxic gluconeogenesis, where protein is converted to sugar because it can't be used in protein synthesis, and now you're getting inflammation in the liver, and we call that steatohepatitis. But we're also getting this buildup of sugar in the bloodstream, we're getting the vascular effects, and we're getting vascular as well as intracellular inflammation, and we call that disease type 2 diabetes, when that system fails. And that, folks, is the carbohydrate insulin mechanism of obesity and diabetes, and it governs fatty liver disease versus steatohepatitis. How do we reverse it? Simple. Simple. The answer to fatty liver disease is to stop eating sugar and starch. And if you adopt a low carbohydrate, high fat lifestyle, a ketogenic lifestyle will resolve fatty liver disease and potentially even non-alcoholic steatohepatitis in 1 week. As a surgeon, as a foregut surgeon, I operate around the liver every single day. I have a whole protocol that I use to get my patients to shrink their liver, to get rid of fatty liver before I operate on them. And I've done over 1,000 liver biopsies looking at fatty liver in those patients. And I know when they've followed the directions or not. Guess what, folks? I know absolutely, biopsy proven, that you can get rid of fatty liver disease in as little as a week, possibly even 3 to 7 days. Just stop eating carbohydrates. And when you do that, the liver may still produce some sugar from gluconeogenesis, but slowly you get into ketosis. And you can get into ketosis in 3 to 7 days, where your body may not be good at doing it, but is using ketones rather than sugar as a primary fuel source. So when you eat a lot of saturated fat, a little bit of protein, and very little glucose, now your insulin levels start to come down, and you develop insulin-sensitive appetite suppression. So it becomes easier to do what we call intermittent fasting, or snacking less often and eating once, maybe twice a day, which gives you a long fasted state, which restores a metabolic state in your liver. And now your fat cells are transporting fat through triglycerides and mostly through LDL back to your liver. That's why your LDL is high, but your triglycerides are low. And now you're manufacturing ketones. And low triglycerides, high LDL, paradoxically, your LDL may actually go up. But as long as your triglycerides are low and your insulin level is low, that is about as healthy and safe as you can be. You do not need and should not treat that high LDL. And that also gives us prolonged and early satiety. So you're eating less and you're feeling full. And most people know this fella. So that the easiest way to resolve fatty liver is to use a low carbohydrate, high fat, mostly animal-based diet, use intermittent fasting of 1, maybe 2 meals a day, use the leptinoids, the hormones that react to saturated fat that tell you that you've had enough to induce early and prolonged satiety, and do frequent bursts of physical activity that encourage cortisol release to release that glycogen and that fat from the liver so that you shrink your liver down. And I know for a fact, proven in our office through 21 years and thousands of patients that you can get remission, biopsy-proven remission of fatty liver disease in 3 to 7 days. And that, folks, is better than any medication. And what are we looking at? I don't care whether you're eating mostly animal products or plant products, but either. You can be more carnivorous or more plant-based as long as you've got a relative mix of micro and macronutrients. But the key thing is to fortify what you eat with fat. Add fat, whether it's olive oil, mayo, butter, eggs, bacon, cheese, nuts, and seeds. Add fat to your food, increase the fat, use intermittent fasting, and eat sequentially so that you allow satiety to happen through the leptinoid process rather than to quantity consumed. Drink lots of fluids and salt in between your meals and with your meals, and make a lot of time for fun things, particularly physical activity, to activate cortisol. That, folks, is called the proper omnicarnivore human diet. And you cannot have non-alcoholic fatty liver disease if that's the way you eat. So the way I manage my patients when they come in, I use a 48-hour, zero-calorie, unlimited hydration with salt fast. So they can drink whatever sugar-free fluids they want to drink, make sure they get a lot of fat, a lot of salt in, but that rapid return to a fasting state just gets the liver rid of that steatohepatitis or steatosis very, very quickly. Then I manage them on a low carbohydrate, high fat, moderate to low protein diet, eating once or maybe twice a day. We use salt and we stop diuretic medications they're on. And if they are profoundly insulin resistant, one of the best drugs we can use, whether they're diabetic or not, is a drug called metformin. Metformin's job is to inhibit gluconeogenesis in insulin resistance. So you stop, you break up the production of a lot of sugar from fat and protein, and you encourage ketosis. It's a way to accelerate this in an insulin-resistant state. So I use metformin often for about 3 months at a fairly low dose. I also, if someone has steatohepatitis, if their inflammatory markers are up, their ALK phos, the gamma-glutamyl transferase, the AST and the ALT, I'm worried about that, and their white count and their CRP, I will add aspirin in as a thromboxane A2 inhibitor of platelet activation to reduce cellular inflammation for a few months. Most of my patients are micro-malnourished. So I will add either a prenatal multivitamin or a regular multivitamin for a few months until their diet is adequate to supply that. I also add fish oil and krill oil, which are DHA and EPA 3 omega fatty acids. Yes, those are polyunsaturated fatty acids, but they are essential fatty acids that have to be consumed and cannot be replaced by crappy fatty acids that get produced from sugar. Then we also use regular physical activity to activate cortisol. Cortisol triggers glucagon to release glycogen and to release some of those ketones from the liver. And in that way, we empty the liver. And physical activity it doesn't matter how intense. It's not about burning calories. It's about cortisol release. We also limit protein intake. We want them to consume animal products. We want them to take in protein, but the overwhelming amount is fat. So I don't like the word protein consumption. I use the word eat animals, eat animal products, because you want your saturated fat and your fat component to be very high so that your fat protects protein from the gluconeogenic pathway. And as your insulin resistance goes away, as you become more insulin sensitive, your protein can then be synthesized. Your amino acids can be used in protein synthesis, not gluconeogenesis. We use a keto mojo type device, which is monitoring blood sugar and ketones to see that GKI, the glucose ketone index, change in patients as they become insulin sensitive. Or if you can, using a continuous glucose monitor is a very useful device. If you really want to know about fatty liver and you can't biopsy the liver, which is the gold standard, liver ultrasound is a very useful way to look at fatty liver, but you've got to have an ultrasonographer that knows what they're doing. And we can then measure liver, lipid, and glycemic blood work and always try to get a C-peptide and insulin that most doctors don't get. Folks, this is not difficult to understand. If you think about alcoholic fatty liver disease, the simplest way to treat it is stop drinking. And if you stop drinking, you are not exacerbating your alcoholic fatty liver disease. Well, folks, exactly the same principle applies to treating non-alcoholic fatty liver disease. No carbohydrates. Done, done, done. This is not that difficult to understand, people. Don't make it more complex than it is. No sugar, no non-alcoholic fatty liver disease. It's that simple. Protect your protein with fat. The proper human diet, folks, is omnicarnivorous. We all desire health and happiness, and that is one of the best ways to get there, through a ketogenic, intermittent fasting diet and a return to an authoritative way of life. Had to throw that in there because I am the Carb Addiction Doc, and I thank you for watching this module. If you have any questions, please access me directly through Messenger on Facebook, through my Instagram account, or through email at robert@jackschildren.com. It's the beginning of the slideshow. I hope this helps. Don't overcomplify, don't make this more complex than it is. It's not difficult. No sugar, no carbohydrates, no fatty liver disease that is not alcohol related.